In this video, I'm going to walk you through how to use SeqMan Engine to assemble and analyze data from the Ion AmpliSeq Cancer Panel, which targets relevant regions across 46 key cancer genes. So the first thing we need to do is set up our assembly in SeqMan Engine. And when you first open the program, you'll see a welcome screen like this. And I'm going to keep the selection of Create New Assembly Project and click Next. Here I need to choose my project type, and I'll select Targeted Resequencing. Click Next. And then I need to specify whether this templated assembly uses a normal workflow or if it's a special workflow. And I'm going to keep the selection of normal. Click Next. Here I need to name my project. And I also need to choose a project folder for my output files to be saved. And I already have that set up, so I'll select it. Then I can click Next. And here I need to specify my template sequence. And for this I'm going to add a genome template package, which is a package of data that we provide on our website for a variety of organisms. And it includes a template sequence, annotations, and associated dbSNP and COSMIC database entries for that particular organism. And you can download these just by clicking on this link here, and that will take you to our website where you can select from one of the available data packages. Now, since I already have mine downloaded, I'm just going to click on the Add Genome Package button. And then I can navigate to the folder that I already downloaded. Then I can click Next to input my sequence files. And here I'll select Ion Torrent as the read technology. Then I can click on the Add button and select the 3 FASTQ files that contain my reads. Now, since these samples have been separated using the Ion Torrent barcode tags, I'm going to check the box next to multiplex data. And then all I need to do is name my samples. So I'll click on Customize Sample Names and then type in the sample names in the group name column. Then I can click OK and Next. And this page allows me to adjust the assembling parameters for my project, but I'm going to leave them at the default settings because SeqMan Engine has already optimized these specifically for an Ion Torrent targeted resequencing project. So I'll click Next, and that's all that's necessary for setting up the project. The final step is to click Assemble, and SeqMan Engine will begin assembling our data. Once the assembly is complete, it can be opened in SeqMan Pro for analysis. And the first thing you'll see here is the project window, which shows the contigs that were created from the assembly, as well as the length and the number of sequences within each contig. And you can sort this list of contigs if you want by clicking on these column headers here. From here, the first thing I'm going to do is open up the SNP report. And I can do that by first clicking on Unlocated Contigs so that all of the contigs are selected, and then go to SNP Report. So you'll see that there are 1,351 SNPs found in this report, and I'm going to do some filtering here, but first I want to point out a couple columns of interest. One is the MID column, and this matches the sample names that I set up in SeqMan Engine before I assembled the data. So this allows me to see which sample contains the SNP. Another column of interest is the feature name column, and you have to scroll over a little bit to see that. And this enables us to know which feature contains the SNP. So you can see, for example, here, there are several SNPs within EGFR. Now, as I mentioned, without doing any filtering, we have over 1,300 SNPs. So to narrow down the list a bit, I'm going to apply a few filters. First, I'm going to select Show Annotated SNPs Only. I'm also going to set the depth filter to 25. And finally, I'll enter 25 for the max coding feature distance, and that is going to show me only SNPs that occur within 25 base pairs, either upstream or downstream from an exon. And now that I have those filters applied, you'll see I'm down to 36 SNPs. Now if I sort by reference position and then scroll through the list a bit here, you can see that, for example, at this position, I have the same SNP occurring in all 3 of my samples. Now if I click on one of the samples, that will open up the alignment view, And I can see the SNP within the context of each individual now by expanding any of these samples. And as a shortcut, if you hold down the Alt key while you click next to any of the sample names, that will either expand or collapse all of them. Now you may also want to look for SNPs that have not yet been annotated, and you can do that from the SNP report by changing this filter here to Show Novel SNPs Only. Now I'm also going to change this filter so that we're only looking at coding change SNPs. And you can see that that brought my list down to 9 SNPs. And just by quickly scanning through, you can see that there's one SNP occurring on each of my 3 samples within the KRAS gene, so that might be one to further investigate. Now, for this size project, analyzing your data in SeqMan Pro is probably going to be sufficient. However, if you have a larger data set and you want to compare SNPs between large groups of individuals, the next step would be to export the data to a RayStar for further analysis. And if you'd like to learn more about that, I'll provide a link at the end of this video to another video that goes through that workflow. If you have further questions about assembling or analyzing data from the Ion AmpliSeq Cancer Panel using SeqMan Engine, please visit our website at DNASTAR.com or contact us at support@DNASTAR.com.